Hello, everybody, and welcome to our first PharmaSource Masterclass of 2025. Microbial fermentation is a foundational technology in biopharma manufacturing, and it underpins the production of so many different therapeutic areas, whether that's biologics, vaccines, antibiotics, and producing key small molecules such as antibiotics and amino acids. And as we'll hear shortly, it's a high growth market driven by the rise of biologics. So I'm pleased to say that on this masterclass, I'm joined by Hervé Ginisti, who is the CSO of GTP Bioways, by Andrea Conforto, Vice President of CDMO Biotech for Olon, and also by Gerald Hoffman, Technology Development Manager at Olon. And so together, the three of them will give you a really good, well-rounded overview of microbial fermentation, share some really interesting case studies, and talk about how you can go through the entire process from early stage all the way up to full commercial manufacturing.
Okay. Hello, everybody, and thank you for joining us in this masterclass. I will introduce a little bit a few slides regarding microbial fermentation and market overview, and then I will leave it to Hervé and Gerald to guide you to our capability in process development and manufacturing. So, with this slide, is just to introduce the microbial fermentation technology. Today is a technology that we are using to make bioactive compounds using microbes mainly. So we use different kind of microbes from bacteria, fungi, and yeast, and we use this technology basically to make different kind of products that could be proteins, recombinant proteins, enzymes, or any other kinds of small molecules. This kind of technology today is used also to make development of advanced biologics, and as well is used in the application of synthetic biology for drug discovery. Yeah, in this slide, it just reported an overview of which kind of product that we can make mainly with the pharmaceutical application using microbial fermentation. On the left of your screen, you can see biotherapeutics and small molecule categories. So with biotherapeutics, we can make a different kind of therapeutic recombinant proteins, antibody fragments, nanobodies. And for small molecule, we are considering all the microbial fermentation. On the right, I report the vaccine. Of course, there are recombinant proteins used for vaccines and any other large molecules that can have a different condition in the pharmaceutical product, like recombinant enzymes that can use the key starting material also within the production of a biotherapeutics or a small molecule. This, with using highlighting also that with this kind of technology, it is remarkable the environmental footprint because again, the raw material that we are going to use here is mainly water, so it's much greener if you compare to the standard traditional methodology used for making pharmaceutical in the past. Now, just let's have an overview of the market for all the products done via microbial fermentation, covering not only pharmaceutical but also food products, food ingredients. Now, in the picture on the right, you can see the trend, expected trend, the market trend to grow from 2023 to 2033. Now, in 2023, we are reporting a revenue generated from microbial fermentation business of over 34 billion, with an expected growth in 10 years over 60 billion. We are speaking about US dollars. The main growth that is around 6% per year is mainly reported for an increase of demand for biologics. And then later we will see which are the potential constraints due to that. Now, focusing on the picture of 2023, we can see that almost 50% of the revenue is coming from Asia, using the microbial fermentation technology. And we expect over the next few years an increase of the players located more in North America. By looking at the application, we can see the great part of the product having revenue in 2023, that is more than 30%, is coming from antibiotics. Actually, if we see also which are the players involved, at least in the picture of 2023, over 40% is covered by and CDMOs. And also we expect that the CDMO will increase their impact on the market in the next few years. Now, in the next slide, we will see mainly which are the reason of the needs of extra capacity for biologics and why there is expected a large involvement of CDMO. First, there is a general increase of the expected demand for biologics. So this means that we need extra capacity. At the same time, to make biologics, the complexity behind is much higher. So a lot of companies are trying to outsource and try to get the expertise from outside if they don't have it inside. In this case, it's expected that CDMO will have huge experience to support the production of those complex molecules. At the same time, also there is a need to keep lower the internal cost. From the other, there will be a different kind of challenges that also the CDMO has to take into account, because making biologics, there is a different kind of complexity compared to the standard API, not only for quality, but also for contamination issue and regulatory requirements connected to it. At the same time, while the production complexity is a topic that will drive company to externalize the production. From the other angles, for the CDMO, is another complexity to take into account, because the process itself usually is longer and much more complex than a standard, I would say, API to produce. And also in this case, there is still competition with traditional or other different kind of technology that can be used for making the products. So, like in the next slide, here we see why microbial fermentation technology is valuable, at least for a certain product categories. We saw already that the environmental impact is lower compared to the traditional chemical synthesis, but also if you compare it with mammalian technology, is more cost effective. The cost of raw material is lower, production cost is lower, and at least also is better, because we can get higher yield and reduce the process time, or at least for the products that can be done in microbial compared to mammalian. Also, from another angle, operation itself can be easily scaled up, also because the scalability is different, it's much more predictable compared to mammalian technology. And at the same time, we also are getting a lot of support from a technology point of view, at least applying CRISPR, artificial intelligence technology for strain optimization and to improve the fermentation process itself. Now, I will leave the floor to Hervé to introduce more what we can do on the services side.
Thank you, Andrea. And so my goal is to speak about early stage development in fermentation. And so this early development in fermentation is placed in a particular context. In most of cases, the projects are carried out by small biotech companies, which are financed by fundraising linked to milestones. Early phase of development are in most of cases associated with new innovative molecule with the aim of reaching clinic as quickly as possible. This often implies that development is driven by decisions such as, if it's work, let's fix this and move to the next step. The main point of attention is placed on a priority target, release a batch which fulfill the specification for first in human as soon as possible. Rather than starting to be part of a quality by design approach. And during these stages, the analytics available are often limited and not finalized, which in fact implies a lack of knowledge during the development. At this stage, several orthogonal methods are necessary in order to highlight the variability, the impact of CQA, and the specific yield associated with each step of the process. Next slide, please. What we learn in over more than 20 years of development at early stage is that the vision of development at this stage is often undergoes the same filter as those that we can see on social networks, with example, the Instagram view with snap filter compared to real view. Development is rarely a straight path, but rather a winning path. Lack of knowledge of the candidate and process are often associated with surprise. The initial development plan is in most of cases revised every week based on the result, and the increasing understanding of the process. Difficulties and bottlenecks that are anticipated were sometimes easier to overcome than expected, and conversely, others appears. Next slide, please. In summary, some key points that I will try to illustrate further through some case studies. Strain is crucial. Verify the compatibility of the strain with further study, compatibility with TDMO, genetic stability, and robustness of the strain over the USP process are essential. Efficiency, quality of the products and processes are often the main drivers in early development, but robustness within the normal operating range has to be addressed. Each new fermentation based project is associated with a specific and unique process, with recipes working for one candidate are often not relevant for another one. And fine and relevant analytics is required to support the development. We need at this stage to build a big knowledge to be compliant with the quality by design approach. Next slide. So in trying to illustrate this key point through case studies, I will start with a short story. Once upon a time, there was a young biotech company that developed a soluble intracellular protein expressed in E. coli. After GSP, USP, and analytic development phase, the process was transferred to the first CDMO, which manufactured the MCB and produced the first batches. For different reasons, including the lack of availability of production slots, the young biotech company decided to change CDMO and once again transferred the process to a new CDMO. The process was transferred and the first technical batch were produced. But at a moment, the CDMO realized that the strain contains a prophage. This information was known from the beginning as the DE3 prophage was introduced during the engineering of the strain. And even if this BL21D strain of E. coli has been used in many existing processes, the quality policy of the new CDMO with regard to management of phage risk prohibit the use of strain containing prophage. So the client has to urgently develop a new strain free of prophages, develop a new USP adapt to the new strain, and verify that this new USP process has no impact on the DSP process, and especially on the, of course, the CQA of the candidate, but also on the level of residual and especially ACP. The story does not say whether they married and had many children, but the moral of the story is that it's necessary to answer the compatibility of the strain with different CDMO and the subsequent stages of the process. Under penalty of losing time, money, and especially knowledge about the process, indeed, the number of runs carried out with this new strain was significantly lower than the one available with the previous strain. Next slide, for example. The following case study concerns the process robustness by illustrating or trying to illustrate the impact of raw material in the variability of process. In fermentation, USP process might either be defined with full synthetic medium or complex medium. In the case of complex medium, batch-to-batch variability might be observed in raw material. This impact is well known from vegetable seeds or peptones whose quality is significantly influenced by the weather or the region of production, but this is also observed in the case of yeast extract, which is supposed to be more stable. Supplier nature is often screened in USP development. However, batch-to-batch consistency might be a concern. Even if it's often difficult to address this question, as only a limited number of batch are available from a same supplier at the same time, often two are available. And in this case, we perform such a comparison with 3 suppliers and 2 yeast extract batches per supplier. The reference one was the supplier A, and the process has been originally developed with this supplier. And we check for alternative. All the alternative supplier that we evaluate outperform the performance of the process, both in final biomass and in specific productivity. However, if the supplier B gave exceptional result on the first batch evaluated, both in biomass and specific productivity, results are inconsistent with the second batch of yeast extract. At the end, we have selected the third one, the supplier C, that show better performance than the reference one, but better batch-to-batch consistency. And we never observed in further batches and scale-up lack of robustness or big variation in the yield of USP. What would have been the result, and what would we have concluded on the scaling up of the process if we had done the development with the supplier B, and what will have been the conclusion regarding the first scale up if we use the second batch for the scale up. Next slide, please. The last case study also focused on the robustness of the process. Once again, a short story. A biotech company has developed a process with another CDMO. All development result and the engineering batches suggested robustness of the process with regard both to CQA and level of residual. However, the first batch, the first clinical batch, even if it was released, show a residual HCP around 95 ppm for a specification that was below 100 ppm. Such a result close to the limit were of course worrying about for the production of future batch. The company come to us in order to refine the process and in order to manage the risk of HCP level. Working on all DSP parameter, but with the constraint of do not change the process flow diagram and raw material that have been used in order to avoid too much change at this phase. We work on holding time, intermediates, conductivity of the different buffer, the washing step, charge of the column, strategy of elution pooling of the 3 chromatography steps that were present in the process, and we succeed in refining the DSP process to ensure robustness regarding HCP elimination and reaching a reproducible value between 30 and 40 ppm. I've chosen this case study to illustrate that often lack of process knowledge during early phase of development is associated with a higher risk of batch failure. We advise our customer to continue process optimization and characterization in order to minimize this risk of batch failure and start to insert in a quality by design approach from development stage, in parallel to their first clinical study that are constrained by fundraising and milestones that they have. Next slide, please. And in conclusion, and to keep the key take-home messages, the first one is strain is key. This is the beginning, and never underestimate the strain of the strain modification might lead to complete process redevelopment and loss of knowledge regarding your process. Never forget that in biologics, the process make the products. Robustness of both USP and DSP are critical, but also focus on scalability of these processes and precise definition of KPI and process parameter acceptable range, and also age of failures. Analytic and documentation within this development is key, and this is the heart of process knowledge. And it required to fit in a quality by design. It is required to fit in a quality by design approach. Never forget that analytical requirements are higher at the development stage than when the process has been validated. After this key message and regarding early development in fermentation, I will leave the floor to Gerald, who will illustrate the next steps and the commercial production.
Yes, hello everybody. Thank you very much, Hervé, for conveying these very important messages, which I think also from an Olon perspective, we are perfectly aligned and in agreement with all the key takeaways that you have been mentioning. What I would like to share with you today is a little bit of our experience and points that we are considering when we are moving from process development or early stage manufacturing to late stage or commercial manufacturing or routine manufacturing as such. And I think the main message that I would like to take you away today from this part about commercial or routine manufacturing is really that when it comes to products that are manufactured by microbial fermentation processes for commercial or routine manufacturing, there is actually no such thing as a one size fits all. What does that mean? This means that if you look on the slide here, we have been putting together a couple of parameters or key factors of products and processes that are made by microbial fermentations. And probably this is nothing new to you, but I think it's very important for everybody to keep this in the back of our heads, is really this enormous diversity of processes and products that are made by microbial fermentation processes. So despite of the fact that on a lab scale that Hervé was just talking about, the microbial fermentation process might look actually all relatively similar, this is actually absolutely not the case once you move into commercial or routine manufacturing, where the manufacturing facilities and the hardware setup required and the process requirements as such can actually be very, very different, and the facility can have an extremely different look and feel depending on what type of product that you are manufacturing. This is mainly due, as you see on this slide, to this enormous range of orders of magnitudes that are spanned by products made by microbial fermentation. These numbers, I think, demonstrate very clearly the very large diversity of products that can be made by microbial fermentation. So what I would like to do is to run you through a couple of considerations when we are moving processes from development to commercial routine manufacturing that are related to these key points that are on the slide here. So the first one is related to the volumes that are involved in microbial production processes. And this is also to explain and make it clear that if you're looking at facilities for microbial products, they can be ranging from relatively small facilities where you can have also just one fermenter of 500 liters or even less, that then typically is installed in a pharmaceutical clean room environment, maybe a grade C or a grade D environment, so highly controlled environments, mainly because just it can be done for these volumes, for these relatively small volumes. But there are also microbial products that are requiring facilities that are just batteries of fermenters that can be 100,000 liters, so 100 cubic meters or even larger. And in this case, of course, typically they are still in closed facilities, but it's becoming extremely complex and challenging if you want to build a pharmaceutical clean room environment around these fermentation facilities. Larger facilities, though, I think the largest aerated microbial fermenters nowadays are exceeding 300 cubic meters. We are arriving at 500 cubic meters or maybe even larger. They are actually typically built even completely in the open environment, which is exposing them to the open environment as such. But as long as you can maintain sterility in your fermenter and that is not interfering with the quality of your process and the product, this is actually not really a concern as such. Talking about these different bioreactor sizes, one of the questions that you should actually ask maybe relatively early on as well is, what is actually the right bioreactor size for my product and process that I'm developing here? The main factor is obviously productivity of the process, meaning the quantity of product that is required per year and the titer that can be achieved at the end of your fermentation process. But there are also other factors that you should consider when picking the reactor sizes. For example, we have experience that if you are running a product in relatively large fermenters, but you are running it only a couple of times per year, then this is obviously giving you a very different learning curve compared to picking smaller reactor sizes where you are running a larger number of batches. If you're relying on the product life cycle, on process optimization and product gains by growing your experience with the process, then you might actually choose to go for a slightly larger reactor size, but that is ensuring you that you can implement your learnings a lot faster compared to if you are running very rarely or relatively rarely fermentations in a very large fermenter where you're getting better economy of scale, but you have limited time for and availability of implementing new process learnings. Talking about volumes, of course, one of the critical points is always the preparation of your nutrient solutions for microbial fermentations, where our rule of thumb is always that if the volumes are getting larger, one should always try to simplify as much as possible the setup. It might be relatively easy to filter a nutrient solution of 1 liter in the laboratory to get a sterile solution. But if then during process scale up, the same solution needs to be sterilized on 50,000 liters, this of course is introducing a lot of complexity and challenges from an operability point of view, because even on 50,000 liters, then you still have to ensure absolute sterility of the solution that is coming out from your filters. And not only bacteriophages can pass 0.2 micron filters, but in case you do not know it, there are also microorganisms that can pass 0.2 microfilters. So this is our considerations that you always have to take very early on in the process. Connecting to the point that Hervé was making about the vegetable peptone and variability from batch to batch, please also do consider when you are finally picking your vegetable peptone of choice, because it's giving you the best performance, also check already very early on, so what is actually the last packaging size that I can get this vegetable peptone in? Because we did have cases where, due to time constraints, we were forced to use hundreds, even close to 1,000, 500-gram packages of certain complex raw materials, because this was the largest packaging size available, and it was not checked. We did not have time to substitute it in the process during the scale-up of the process. So if you need 50 grams during lab scale development, that is not a problem if you have 500-gram packages. But if then for scale up you need 500 kilograms of the same material, this can become very complicated. Moving away from volume a little bit, but touching the next two factors that are also connected to process reliability and robustness. We are mentioning here the molecular size as well as the commercial value of products that are made by microbial fermentation. And these two, as you might also probably know, are actually directly correlated. So typically, if you're going for large molecules, the value of these molecules is very much increasing. And the difference between the process control and the robustness and the reliability that you need for implementation of large molecule fermentation processes is actually very different from small molecules. Obviously, if you're producing a small molecule, at the end of your fermentation process, you can 100% characterize your final product. For example, penicillin, which was the first major biotechnological product made by microbial fermentation. You can, at the end of a fermentation process, characterize your penicillin molecule 100% make sure that every atom is in the right position where it's supposed to be. However, if you are making a protein that is 500 kilodaltons, meaning 500,000 grams per mole, then even with all the methods that we have available nowadays, it's becoming basically impossible to 100% characterize your product. And this means, as Hervé was mentioning before as well, that in these cases, what is nowadays always said is that your process is actually becoming your product, meaning the only way to guarantee that the product that you are making in the end is always the same is by having very tight process controls around your process. And this obviously is starting from your fermentation process, where for the first time you are making your API, your large molecule. What we routinely make for these processes is that we are using an approach called the failure mode and effect analysis that allows you to quantify and identify the critical parameters and the critical parts of your process. And there, for example, you might see that for specific cases, a glycosylation of a large molecule might largely depend on which dissolved oxygen concentration you have in your process. And if this is identified as a critical parameter, you might consider that you don't want only one probe that is giving you the information around the dissolved oxygen, but maybe you want 2 or 3, because as much as these probes have been improving over the last years, they still do break. And in these cases where they are highly critical for your process, we always do recommend to have redundant controls. So in case one probe is failing, that you can still control your process within the given parameter ranges. Saying all of this, I would also like to share with you in the next slide a couple of very short case studies that are describing a little bit of our real-life experiences with microbial fermentation processes, where the first case is a very typical case, I would like to say, where we have been implementing the manufacturing process of a nanobody, which is a very small fragment of an antibody, which in this case was produced in Pichia pastoris. And this is really one of the classical cases where we have been taking in the process on the laboratory scale, perform process development and scale it up. And as this was not a pharmaceutical product, but a product that is actually used in agriculture, we actually managed to do all of this within approximately half a year, 6 months. And after approximately a year, we managed to implement the process very stable and robust on commercial industrial scale after running a consistency campaign and also validating the process on industrial scale, which is ensuring you that you can run the process repeatedly and achieve the quantity and quality of your product that you want to achieve. The second case that I would like to share with you on the next slide is a very different case. In this case, the request from a customer was coming to implement a process that was already running at commercial scale. And as in this case, there was already a lot of experience of this fermentation process at industrial scale, we decided to directly implement the fermentation process on industrial scale and actually managed to provide the customer with product for the market to be used in the market, meaning commercial product, within 3 months after the project initiation. After this first 3 months, the project was, however, far from finished. As in this case, we decided together with the customer to use what I've been describing before. We have been using smaller fermenter sizes, in this case, where each fermenter was producing approximately 20,000 liters of fermentation broth. But as the purification capacity was expanded over time, we actually have used a setup where we ran 3 fermenters in parallel, meaning every fermenter discharged, or the DSP plant actually received every 48 hours 20,000 liters from one fermenter. So means we had a very high cadence, a very high frequency of batches going through in a campaign. And we were leveraging this to systematically and structurally fine-tune all the parameters of the process to optimize it, and over time achieved very large improvements of the processes. An example of this, for example, was that in this case, we were killing off the microorganism that was a GMO in the fermenter at the end of the fermentation process. And by fine-tuning the conditions in the fermenter during this killing off step, which were the pH, the temperature, but even the dissolved oxygen during this killing off step, we managed to avoid product degradation, which typically occurred during this microbial inactivation step, and managed to significantly improve the output of the fermenter for every run. So these are just two very short examples that we wanted to share with you. But again, I think the main message is that for commercial and routine manufacturing, we do see that every specific product and molecule requires a very specific production facility setup and a very specific implementation process optimization program until you're arriving at a commercial routine manufacturing process. And with that, I would like to pass it on again to Andrea for some last words.
Thank you, Gerald. So now in these next few slides, I will just guide you through the services that both GTP and Olon Biotech can give to the microbial fermentation field. So starting from GTP, as already also anticipated by Hervé, we can support you in different stages of your process development, starting from discovery, moving to the clinical development up to GMP production for an early clinical stage. In GTP, we have a very large experience, different kind of products, mainly large molecule like recombinant proteins, antibody fragments, enzymes, nanobodies, both in process development and GMP manufacturing for early clinical stage. We have experience and capability to support you from strain development up to process development in both in fermentation DSP using design of experiments, DO experience. And also we can support you also in the analytical part. Therefore, the support for the GMP production for early clinical stage, we have a fermentation capability up to 350 liters. Moving to the next slides, we will just now show you what we can do, vice versa, from Olon Biotech side in the microbial fermentation field. Also there we have pilot lab capability that we can support for tech transfer and also for some process improvement, and as well for piloting up to 3 cubic meters. While for clinical and commercial production, we can support with different kind of capability in our microbial fermentation sites. We have 2 microbial fermentation sites located in Italy with a total fermentation capacity of 4,500 cubic meters, distributed over more than 60 fermenters with different fermentation sites. The smaller one that we are using for GMP manufacturing is around 15 cubic meters, up to maximum one. The bigger one is 255. In between, we have fermenter sites from 35 to 50, 60, 100 cubic meters. We have quite large experience in both sites with different kind of products. Also, they're making small molecules and large molecules like enzymes and therapeutic and proteins. Also, there we have more than I would say more than 50 years experience, because microbial fermentation started like 70s. And also we have currently ongoing, looking just only at the CDMO portfolio, more than 20 projects ongoing currently in both sides. Now moving to the last slide, we can give now with this last slide, the next one, right? Thank you. How we can support you from the full process, from discovery and early stage development with the GTP up to clinical and commercial manufacturing in Olon Biotech. We have quite large recognized experience in both companies in supporting and making different kind of proteins, enzymes, using different kind of strains, both from fungi and different kind of also bacteria. Also, we can support in the technical transfer all the process. So basically, everything will be done internally between GTP and Olon Biotech, and we can be very quick in implementing the process at large scale. So now I hope that with these slides we gave you an overview how we can support you, basically from early clinical up to commercial with very small scale up to very large scale to be done everything in GMP. And now in this last slide, just we like to give you some contact details in case you would like to contact us to get more information on what we do and how we can support you. Thank you for joining us.
Thank you very much. Grazie mille. Thank you, Andrea. Thank you to Hervé and Gerald for such a fascinating deep dive into this really important area. I thought the stories that you shared, the case studies, really brought it to life. And I think, Gerald, the quote you said about how your process is your product is absolutely key to everything that we've talked about today. We do now have time to do questions and answers. So if you're dialed into this call, you're obviously interested in the topic. Now's your chance to put your questions to our subject matter experts here. You can do that by adding them to the chat, which you can see at the side of the screen. If you add them to the chat, then I can put them to our experts. We have two that's come in already. Actually, Tamina and Dante, I think you're basically asking a very similar question about process development and documentation. So Dante, you said it's very complicated to understand how a new process can be used at a large scale. Process requires specific activities to ensure the quality of the product. Can you suggest documentation and scale up activities? And Hervé, maybe this is a good one for you, please.
Yes. So first, the documentation describes what has been achieved and done during the development. So more than the documentation is the question is, what are the main questions that we have to ask in early development in order to answer a good scale up? Of course, there is some key parameters that are well known. Of course, description of oxygen transfer rate and so on are key parameters in order to ensure the scalability of the process. But if I have to summarize, I think that the projection to the commercial scale and the size of the batch at the commercial scale will be key because it will drive all the anticipation. For example, when you go to 100 of cubic meter fermenter, you often in the same time reach the limit of chromatography that has been developed during the thing, because the media and the size of the column have the limit, and often the productivity is so high that you have to run multiple runs on your DSP. So from one USP batch, you have to make different cycle on the column or different DSP processes to reach the product at the end due to contrast of size. In this example, it will be critical to describe the stability of intermediate in order to know or to be sure that you can run, I don't know, 10 cycles on your chromatographic column, or you will have to change of technology and to find an alternative in order to ensure scale up. And often this is thing, of course, when you are a young biotech company developing your first product, is really difficult to make some projection at the commercial scale. But for me, it's essential in order to answer scalability and avoid a loss of time in between in order to solve scalability troubles.
Great, thank you. Is there? I'm wondering whether it's something that we could share with attendees after this event. But is it possible to have a template? I mean, I appreciate that every project's different, but is there like an overall process development template or framework which maybe we could share with people after the session?
I think it will be difficult, but we can try to make such kind of summary. I think that we know that, and I'm sure that Gerald will totally agree. And we know that centrifugation and chromatography are often the main difficult thing to scale up and might be the main limitation at large commercial scale. Of course, it's completely different if you deal with an injectable, highly potent for a small market, or if you are dealing with compounds that are addressed to orally to a large portion of the population. The scale are completely different and the limitation are different. But yes, I can we can try to summarize a flow scheme or a decision chart in some.
I think that would be helpful. At the
box.
very least, to everyone on the call, we will be sending you a copy of this video afterwards as well, so you can also watch that back. Maybe Gerald, maybe if I could bring you in, interested to get your perspective on that question about process development. I think it's interesting in the poll that we ran at the beginning. The majority of people, so 27%, said that that's the stage that they're at, is the process development and optimization stage. So any advice you could share on that would be helpful, please.
Yes, no, it's of course, as Hervé is saying. So unfortunately, as I've been trying to point out before as well, for microbial product, it is really difficult to provide templates because every product and process, it has its own story, its own limitation, its own requirements. But of course, we can try to set something up. I always just saw there was the last question that was coming in around the processes, the product, which is also connected to this. And of course, it's very important during process development and then also during commercial to have a proper documentation system where you are really tracking and tracing everything that you are learning about the process. Some people are not realizing it, but if you're running a process on lab scale, where, for example, in a purification process, typically you do not work during the night as well, you actually automatically already have a lot of information of potential holding points on larger scale for routine manufacturing afterwards, because you know that if I stop here and put it in the fridge in the evening and take it out the next morning, my product is still there and I can continue using it. So collecting all this information is actually very critical and useful to ensure proper and optimized process development and keeping track of things that are important.
Great. And then we've got a question also about cross-contamination. So is it possible to produce API products such as antibiotics and proteins in the same plant?
Yes, I can also take the short answer nowadays is yes. It was not possible. Actually, it was not allowed by FDA regulation or EMA regulation, I think until something like 10, 15 years ago, where it was required to segregate, in particular, beta-lactam antibiotics, because they are conferring a very high risk if you have people that are allergic to it. So even if you find traces of products of beta-lactam antibiotics in another product, this can give allergic reactions and potentially kill the person. So that was the base for this whole discussion. But regulations have been relaxing on this over the last 10, 15 years, and nowadays it is allowed to do it. It is not very common to do it, I have to say, and you still have to demonstrate that there is absolutely no carryover from your beta-lactam antibiotic to any other product. But as such, it is allowed and feasible to do it. Of course, we're also talking mainly about antibiotics being small molecules. We're using solvent-based processes versus proteins being larger molecules and typically using water-based processes. So there isn't a big overlap between manufacturing facilities for these two types of microbial products as well.
Great, thank you. And unless anyone's got any further comments to add in, I'd just like to throw one question at the pair of you, please. And that's, you've obviously shared a lot of information here. I wonder if there's just one piece of advice which you could share, maybe just one, if people take away nothing else from this entire session except for one thing, what would that be, please? What's the most important thing for you? And maybe Hervé, if we could start with you, please.
I think that continue the development or the characterization on the continue to understand your process, even if you have reached the first goal, that is your first batch can go to clinic, is really key if you want to avoid any further trouble and loss of time.
Great.
Yeah,
thank you.
I think my point is very much connected to this one, which is try to learn as much as possible about your specific product as early as possible. the earlier you understand what are the specific requirements for your product, and the more you are diving into the specifics, the faster you are going to arrive at commercial manufacturing.
Thank you, Gerald. Andrea?
Yeah, what I would like to recommend is, in case you think that you need external capacity, be in touch with the CDMO ASAP. Also, while you're making, if you prefer to make your own development in-house, start talking with the CDMO, because again, knowing what you can do later in production, for early clinical production or for commercial, is better, because also the CDMO can support you in the development, even of not making the services like we can directly, but also provide an idea how to make the process later scalable at industrial scale. Also can help you a lot in transferring the process to manufacturing. So don't hesitate to contact, to speak with the CDMO, even if you don't need capacity on short time. It will help you anyhow in your process development.
Wonderful. Thank you so much. So all that's left for me now to say to wrap up is, if you want any more information about this, you can see the contact details there on the screen. There's olonspar.it, gtpbioways.com, with their email addresses there if you want to reach out to the team for more information. From us at PharmaSource, we will be sending out more on this as well, the recording of this webinar with an article to accompany it. So thank you very much for joining us. I hope it's been a useful session for you.